Good afternoon, students. Yesterday we discussed about transposable genetic elements, also called as transposons. Okay, so what are transposons? They are mobile genetic elements capable of inserting themselves at a different site within the same chromosome or maybe in a different chromosome. Now, they are ubiquitous in nature and they are found in both prokaryotic as well as eukaryotic organisms. They are normal, right? Now, and if you look at types of transposable genetic elements, there are two basic types. Number one, IS elements, and number two, transposons. So, yesterday we discussed about insertion sequence, that is IS element. What are the characteristic features of IS element? IS element is the simplest type of transposon having a gene which codes for a protein transposase required for transposition, required for mobilization and integration of insertion sequence into the target site. Okay, so that is one essential feature of IS element. The second essential feature of IS elements is at both the terminal ends, that is at the right terminal and left terminal, it has a inverted terminal repeat. Okay, so insertion sequence has a gene code encoding transposes protein and inverted terminal repeats at both ends. So it's the simplest insert type of transposon. And how it gets how the transposition of IS elements takes place? The IS element is first duplicated and the transposase enzyme creates a staggered cut in the target site. Okay, it cuts the DNA at target site. What is the target site? The site where the transposon or IS element is inserted. So staggered cuts, as we discussed yesterday, staggered cuts are made in the chromosomal DNA, right? The chromosomal DNA separates and the IS element is inserted between the staggered cuts. Okay, and now wherever there are staggered cuts, single stranded ends are generated and the single stranded gaps are filled by DNA polymerase enzyme, this gap as well as, but at the staggered cuts there are direct repeats. Okay, and the single stranded staggered cuts are converted into double stranded ends by the DNA polymerase enzyme. Okay, and the gaps are sealed by ligase, and this is how duplicated target site sequence. Okay, and in between there is a insertion sequence inserted in the target site, at the target site. Okay, now let us discuss about transposon proper. Similar to IS elements, transposons are transposons are similar to IS elements but structurally more complex and have additional genes as compared to IS elements. There are two types of transposons: composite transposons and non-composite transposons. Okay, what are composite transposons? They carry genes. Okay, say for example, they may be having antibiotic resistance genes, and on both sides they are flanked by IS elements. That means in the center there will be a antibiotic resistance gene. Say for example, tetracycline resistance gene in the center, and at both the sides, that is at the left corner as well as at the right corner, there will be IS element. Okay, so centrally placed gene like antibiotic resistance gene, and at both the corners there will be IS element. That means if you look from left hand to right hand, IS element, antibiotic resistance gene, and again IS element. Okay, say for example TN10 means transposon 10 is 9.3 kb and it includes 6.5 kb kilobase pair of central DNA. Okay, and the central DNA is tetracycline resistance gene. So central DNA is 6.5 kb in TN10, transposon 10. Total length of TN10 is 9.3 kb. Central region 6.5 kb. The 6.5 kb is a gene having tetracycline resistance. Okay, it includes a gene with tetracycline resistance, and at both ends 1.4 kb of inverted IS elements. What is the role of IS elements here? They have transposase gene. So they will supply both transposase enzyme and inverted terminal recognition sequence. Okay. Transposase enzyme carries out its work only when it identifies inverted terminal repeat. Okay. So every transposon should have transposase gene and inverted terminal repeat. So here the IS elements are flanked at both the ends. Okay. So complete IS element is present at right terminal end, left terminal end, and in the center antibiotic resistance marker gene is present in composite transposon. Okay, look at this diagram. This is TN10. It is a composite transposon. Right. If you look at the center of the transposon, 6.5 kb, that means 6500 base pairs. See, it is the central part. Okay, and in the central part there is a gene which offers antibiotic resistance. Here tetracycline resistance gene is present, and at both the left corner as well as at the right corner there are inverted IS elements, complete IS element. Okay. If you look at both the IS element, they are having 1.4 kb length, that is 1400 base pairs. Right. If you look at IS 10L means on the left side of transposon 10, and IS 10R means on the right side of the transposon. Okay, 10. Now if you look at any of the two IS element, the central part, the yellow colored part shown in the center is the transposase gene, which will code for the protein transposon, and the red color ends of this IS elements are inverted terminal repeats. Okay. So this is a composite transposon having in the center this is a composite transposon having in the center 6.5 kb gene, wherein there is a gene called as antibiotic resistance marker gene or tetracycline resistance gene. At both left end as well as at right end there are complete insertion sequences. Okay, so this is a composite transposon made up of two IS elements and a centrally placed antibiotic resistance gene. Is that clear? This is a composite transposon. Now another type of transposon is non-composite transposon. The non-composite transposons have genes that might be genes for antibiotic resistance. Some additional genes are also present in non-composite transposons. Okay, apart from antibiotic resistance genes, some more genes are also present, but they do not terminate in IS elements. That means at the left terminal end or at the right terminal end, IS elements are not complete, IS elements are not present. Okay, so what is the difference between a composite and a non-composite transposon? In a composite transposon, centrally placed DNA has antibiotic resistance gene, and at both the terminal ends there are IS element. In non-composite transposon, centrally placed genes are there. Antibiotic resistance gene is there. Apart from antibiotic resistance gene, other genes are also present. But at the terminal ends, complete IS elements are absent. Okay. Ends are non-IS element repeated sequences. Non-IS element. That means repeated sequences are there. Repeat sequence are there at both the terminal ends, but they are not IS elements. Is that clear? Say for example, TN3, transposon 3 is the example of non-composite transposon having length 5 kb, that means 5000 base pairs, 5 kb, okay, with 38 base pair, inverted terminal repeat. Okay, there are only inverted terminal repeat. Complete IS elements are not present at right or left terminal. What is present at the right or and left terminal? Inverted terminal repeat. Only, say example we have taken TN3, so 38 base pair inverted terminal repeat. Is that clear? And in the center there are three genes. Number 1, beta BLA gene, which produces the enzyme beta lactamase. Then TNPA, that means transposase gene, because IS elements are absent, but transposase enzyme is needed for transposition. So transposase enzyme will be required. So who is going to prepare that? In the absence of IS element, in that case, in the center itself, the non-composite transposon in the center has three different genes: beta-lactamase gene, transposase gene which produces enzyme transposase, and resolvase gene which helps in recombination. Okay, so there is TNPA, that means transposase enzyme producing gene, TNPB means resolvase enzyme producing gene, and say beta lactamase gene or antibiotic resistance marker gene will also be present in the center. And at both the corners, what will be present? Inverted terminal repeat. So this is the diagram of TN3, which is a non-composite transposon. So you can see in blue color there is TNPA gene. Okay, so this produces enzyme transposase. There is TNPB gene which produces enzyme resolvase. There is BLA gene which produces beta lactamase enzyme. Right. Apart from that, at the right corner as well as at the left corner, left inverted repeat 38 base pair, and at the right corner, right inverted terminal repeat. Okay, at the right corner, right inverted terminal repeat. Right. So at both the corners, IS elements are absent, so it is called as a non-composite transposon. Right. So centrally placed three or more genes, and at the two corners, right, left inverted terminal repeats. Now how the transposition takes place? Let us understand the mechanism or model of transposition. Number one, replicative transposition. What happens in replicative transposition? The IS element or the transposon duplicates itself. Okay, that means it produces a copy of itself, so that the element which is transposing, the element which is moving to the target site, is the copy of the original transposable element. Okay, replicative transposition mechanism is the one in which the transposon or the IS element duplicates itself, and the duplicated copy moves to the target site when the original copy, transposon or insertion sequence is there in the initial site. The resolvase enzyme is involved in site-specific recombination between two transposons present as direct repeat and in a co-integrate structure. Now there is another mode of transposition which is called as non-replicative transposition. What happens here? The transposable element moves as a physical entity directly from one site to another and is conserved. That means from one site where it is originally present, it will jump from the site, or it will move from the original site and go and get inserted at the target site. That means at one place it is not there, and at the target site the transposon will be present. So the transposable genetic element moves as a physical entity. It is not duplicated. It directly moves from one place to another and it is conserved. Okay. And there is another conservative transposition. It is a type of non-replicative event only, in which element is excised from the donor site and inserted into the target site by a series of events in which every nucleotide bond is conserved. Okay. Though it is a non-replicative event, that means the transposon is not replicated. It moves from donor site to the target site, but every nucleotide bond is conserved and hence the copy can be synthesized. Right. Now here you can see replicative mode of transposition. Now this is the donor. Okay. The first, this is the donor. Yeah, you can say this is the initial position of the transposon. And this is the recipient cell. This is the target site. Okay. This is the target site. It has to be cut. The recipient position has to be cut at the target site. Now, this is a kind of replicative mode. Right. So, the donor remains unaltered. Right. The transposon makes a copy of itself and the copy is inserted at the target site. Okay. The donor DNA, the transposon or insertion sequence makes a copy of itself and the copy gets integrated at the new site, but the donor remains unaltered and the recipient gains a copy of transposon. Okay, so this kind of mechanism involves resolvase enzyme as well as transposase enzyme. Transposase will behave as restriction enzyme and cut the target site in the recipient. Okay, and resolvase enzyme will help in transfer of DNA from original site to the new site when the donor DNA or transposon is actually duplicated here. The replicative transposition creates a copy of the transposon which is inserted in the target site. The donor site remains unchanged. So both the donor and recipient have the copy of transposon. This is one method which is called as replicative mode of transposition. Now this is non-replicative mode. This diagram shows non-replicative mode where only transposase enzyme is required. Okay, the transposase enzyme allows to move the transposase enzyme allows the movement. So this is the donor, okay, and the colored one, that is red orange colored one, is the transposon. It gets removed or excised from the donor. It breaks off from the donor and gets integrated at the target site in the recipient. Okay, or you can say it moves as a physical entity from donor position to insertion position. So from one point it is lost and from at another point it is integrated. So the donor you can say loses the transposon and the recipient gains the copy of transposon, and only transposase enzyme is required. This creates a gap or a break in the donor site, and it may be lethal because it may lead to deletion type of mutation, and in this case the repair of the donor, the repair of the host is required. If not repaired, it may lead to mutation because there is a gap is created, a break is created at the donor site. Okay, this is the third type, which is a kind of non-replicative but conservative mode. So you can see from the diagram, the transposon from the donor directly gets integrated into the recipient site, which is similar to a non-replicative mode of mechanism here, but the sequence is conserved. Every nucleotide bond is conserved. There is no loss of nucleotide bond. Okay, right. So this is called as a conservative mode of replication. So that is all about transposons, composite transposons, non-composite transposons, and the mechanism of transposition, which may be by replicative mode, non-replicative mode, and number 3, non-replicative conservative mode. Okay, so that is all about transposons.